Assalamu alaikum dear aimers, hope so you all are doing well. I am Ariba Nadeem and today the topic we are going to study is dietary lipid and metabolism. Be clear about the topic that we are going to study about the dietary lipids. It means that the lipids which have been taken in the diet. And metabolism, you all know that in metabolism we will study about two processes, catabolism and anabolism. Firstly, we will study how to synthesize a lipid and then we will study how to break a lipid and get energy from it. But in this chapter, we are only going to study about the metabolism of dietary lipids. So what are the sources of dietary lipids? So there are four important dietary lipids we take in our diet. Firstly, it is TGs, then we have phospholipids, then we have cholesterol, and then we have cholesterol ester. Do remember that the lipid intake should be approximately 78 grams per day. Okay, so you have to take 78 grams per day lipids, and out of which 90% are the TGs we take in our diet. So 90% are the TGs. So from which diet do we take TGs? Be clear about it. Like TGs are present in red meat, they are present in dairy products, they are present in butter and margarine. So all these stuff contains triglycerides. Similarly, phospholipids are also present in your red meat. It is also present in egg yolk and it is also present in milk. Then we have cholesterol. Cholesterol, you all know that two important sources of cholesterol are egg yolk and second one is red meat. So these are two important sources of cholesterol. And then we have cholesterol ester, means the cholesterol which is bound with fatty acid. It means cholesterol plus fatty acid, and then they have an ester bond between them, so we call it as cholesterol ester. Do remember 90% are the TGs and these all make only 10% of the lipids in our diet. Okay, now what are the steps of lipid metabolism? Firstly, we are going to study about how we take lipids and how these lipids and then metabolize, how they are metabolized in our body. So firstly, the first step in metabolism is digestion. Of course, we take the lipids and the first step is that the lipids get digested in our body. So first step is digestion. Second step is the absorption of lipids. Absorption. And third step is secretion. And fourth and final step is utilization of the lipids. Okay, so there are four important steps: digestion, absorption, secretion and utilization. So suppose you have eaten the red meat. Okay, so that red meat contains TGs, that red meat contains phospholipids, that meat contains cholesterol. So what is the metabolism of that lipids present in the red meat? Firstly, that lipid would be digested, then it would be absorbed, then it would be secreted in the blood, and then it would be utilized by muscles and adipose tissue. So now the next step is we're going to look at the sites where these steps are taking place. Firstly, we have digestion. The step of digestion basically takes place in stomach and small intestine. So we have two sites for digestion. First is stomach and second one is small intestine. Then the second step is absorption. This step takes place in small intestine. Next step is secretion. When the lipids are absorbed in small intestine, later they are secreted into the blood. And the final step is utilization, where that lipids will be utilized. They will be utilized in muscles because the muscles will oxidize the lipids and will get the energy from it, and they will also be utilized by adipose tissue because adipose tissue will store those lipids. Okay, so these are the sites for the metabolism of the lipids. Now be clear about some bonds which we will study in whole lipid metabolism. And those important bonds, first and foremost important bond is the ester bond. So do remember what is the ester bond. I'm going to make ester bond over here. It is carbon, double bond oxygen, then we have R, and here we have O and then R. Please do memorize this bond. This is an ester bond, and do remember specifically this is the ester bond. whenever you are going to synthesize lipid, or especially when you are going to break the lipid, always break this bond. You will realize its importance when you will be doing the beta oxidation or the steps where you are going to break the ester bonds. Because what do the students do? Sometimes they break the ester bond from here, sometimes they break the bond between oxygen and alkyl group, but you don't have to do like this. Always remember, you have to break the bond between carbon and single bonded oxygen. So this is the ester bond. Second important bond over here is the acyl group. So students get confused about the acyl group. Simply carbon double bond oxygen R. This is simply the acyl group. Wherever you have, like later on we will study about monoacyl glycerol, diacyl glycerol. So this is simply carbon double bond oxygen with any alkyl group, that is simply termed as alkyl group. And we also we will also study about one more bond, which is carbon double bond oxygen R, nitrogen bonded with hydrogen, and then we have an other R group. So this is basically the amide bond. So do remember that in ester bond, we have one functional group carbon and two functional group oxygens. And the bond we have to break is between carbon and oxygen. In acyl group, the bond is between carbon double bond oxygen and then we have R group. And then the amide is carbon double bond oxygen R, it's same like that. But what you have to do over here is that nitrogen is bounded with hydrogen and R. So what we have studied till now is, we are going to study in our chapter dietary lipid and metabolism. It means we are going to talk about exogenous lipids, because lipids are also synthesized within your body. We're not going to talk about the metabolism of the lipids which are synthesized in your body. We are going to talk about the metabolism of lipids which have been taken from the diet, and approximately the dietary intake of lipid per day should be 78 grams. Out of these 78 grams, 90% are the TGs or triglycerides, which are saturated or unsaturated. We will talk about it later. Then we have phospholipids, cholesterol and cholesterol ester, which makes up 10% of the dietary intake. TGs are present in your butter, margarine, red meat. Phospholipids are present in your red meat, egg yolk, milk. Specifically, you have to remember about cholesterol, that it is only present in egg yolk and red meat. And then we have cholesterol ester. It means the cholesterol bounded to fatty acid. We have an ester bond between them. Then we talked about the steps. The steps are digestion, absorption, secretion, and utilization. Then we talked about the sites where they are going to happen, where these processes are going to happen. Digestion starts in stomach and ends in small intestine. Absorption occurs in small intestine. Then the lipids are secreted into the blood, and when they have been broken down into fatty acids and cholesterol, then they are utilized by muscles for getting the energy and adipose tissue to get stored. Then we talked about bonds, the ester bond, the acyl group, and the amide group. Okay students, now in this video we are going to study about digestion in detail. Now what is meant by digestion? Digestion simply means to convert polymers into monomers. So it is the breakdown of polymers into monomers. For example, if we have a sucrose intake, then that would be broken down into glucose and fructose. Similarly, in case of lipids, the lipids will be broken down into its simpler units. Okay. Now, what were the dietary lipids we studied about? We studied about TGs, triglycerides. Now, triglycerides will be broken down into fats and glycerol. Similarly, we studied about phospholipids. Phospholipids will be broken down into glycerophospholipids. We'll study about it later. Glycerophospholipids. Similarly, if we have cholesterol ester, that would be broken down into cholesterol and fatty acids. Okay, free fatty acids. Okay, also remember what is the difference between fatty acid and a free fatty acid. Fatty acid simply means fatty acid. What is meant by free fatty acid? Free fatty acid, wherever the word is written free fatty acid, it means it is the fatty acid which has been obtained from the breakdown of lipid. Okay, it means that some lipid has been broken down and the fatty acid which was released from that lipid, that would be termed as free fatty acid. Okay, like we broke cholesterol ester and then the fatty acid was released, this would be termed as the free fatty acid. Now these are the polymers and these are the monomers. It means that digestion has been completed. And I told you earlier that the digestion occurs in stomach and small intestine. It begins in stomach and it ends in small intestine. Now we are going to look at the enzymes which are needed for the digestion. You know, every metabolic process needs enzymes, and enzymes work in presence of water. Now, what are those enzymes which are really used in digestion? There are three basic enzymes which are used in digestion. First enzyme is termed as lingual lipase. Lingual lipase. Second enzyme is gastric lipase. We will all enzymes will end in lipase, means lipids breaking enzymes. And the third enzyme over here is pancreatic lipase. Okay, pancreatic lipase. Lipase means to break, the one which is breaking the lipids. Now we will study each of the enzyme in detail. Now, what is meant by lingual lipase enzyme? What is the action of lingual lipase enzyme? As the name goes, lingual suggests tongue. Okay, lingual means tongue. This lingual lipase is basically a minor salivary gland. As you know, there are three major salivary glands: the submandibular, the sublingual, and the parotid. But this lingual lipase, it is a minor salivary gland. So do remember, it's a salivary gland, and it is released from both sides of the oral cavity behind the tongue. This gland is present. This basically it is released from von Ebner's gland. Let me write here, von Ebner's gland. This von Ebner's gland is a minor salivary gland. Major salivary glands are parotid, sublingual, and submandibular, but this von Ebner's gland is a minor salivary gland, and this is present on both sides of the oral cavity behind the tongue. So this lingual lipase is released from this von Ebner's gland. Now do remember that this lingual lipase is secreted in oral cavity, but it gets activated in your stomach. So its activation is in stomach. Why it gets activated in stomach? It is because it acts in low pH. It acts in pH approximately 4 to 6. So this lingual lipase secreted into the oral cavity, gets straight into your stomach and gets activated in your stomach, performs its action into the stomach. So always remember that the digestion of lipids begins in stomach. Never say that the digestion of lipids begins in oral cavity, because this lingual lipase is not activated in oral cavity. It is secreted from the oral cavity and then gets activated into the stomach. So you will always say that digestion of lipids begins in stomach. Now we have the next enzyme, which is the gastric lipase enzyme. This gastric lipase, wherever the word is gastric, be clear, it is related to stomach. Okay, so gastric lipase, it is released from chief cells of stomach. You know, chief cells, the one releasing the pepsinogen, converting the pepsinogen to pepsin. So chief cells are those which release pepsinogen, and chief cells are those which release this gastric lipase enzyme. Always try to connect the things. So whenever you are studying chief cells, do remember they also secrete pepsinogen and they also release gastric lipase enzyme. And pancreatic lipase, that is simply secreted from the exocrine portion of the pancreas. The endocrine portion is releasing the insulin and glucagon, and the exocrine portion of pancreas is releasing the pancreatic lipase enzyme. Now there are some characteristics which are common for lingual lipase and gastric lipase. So I'm going to write over here. Firstly, first point you have to remember for both of these enzymes, that these enzymes work in acidic pH. And stomach has acidic pH, which is approximately 4 to 6. So these enzymes work in acidic pH. Second most important point is that these enzymes are responsible for the breakdown of medium or short chain fatty acids. Medium or short chain fatty acids. You know what are the short chain or medium fatty acids? The fatty acids having the carbon R chain length is less than 12 carbon. Okay. So whenever the fatty acids have length less than 12 carbon, that is what the lingual lipase and gastric lipase, they are going to be in action, and they are only responsible for the breakdown of medium or the short chain fatty acid, the fatty acids having the length of approximately less than 12 carbon atoms. Now, what is the third most important point? Third most important point is they are more functional in children. Why they are more functional in children? Because do remember, children have low quantity of this pancreatic lipase. Okay, so major function is performed by lingual lipase and gastric lipase. But in case of adults, the main enzyme for the breakdown of lipids is pancreatic lipase, while the minor enzyme is lingual lipase and gastric lipase. Do remember this point, that the lingual lipase and gastric lipase is more functional in children because they have low quantity of this pancreatic lipase. Second point, why? Because the children, they are infants, they are more dependent on milk, and milk contains short and medium chain fatty acids. Milk is rich in short and medium chain fatty acids. So this enzyme, both of these enzymes, they are more functional in children. Also, this enzyme, the load on these enzymes increases in case of patients, cystic fibrosis patients. We will study about this disease later. But this is some kind of disease in which pancreatic lipase is not released. Okay. So that's why the load on lingual lipase and gastric lipase increases. So this enzyme, both of these enzymes, are more functional in children and the patients suffering from cystic fibrosis. Now the third point is that these enzymes, I told you that they are more functional in adults and children. Okay, what we studied till now, we studied about the digestion, which simply means to convert polymers into monomers. TGs will be broken down into fats and glycerol. Phospholipids will be broken down to glycerophospholipid, and cholesterol ester will be broken down to cholesterol and free fatty acid. I told you the difference between fatty acid and free fatty acid. The enzymes used over here are three: lingual lipase, gastric lipase, and pancreatic lipase. Just revise with me. Lingual lipase will be secreted from von Ebner's gland, which is a minor salivary gland, and it is present both sides of the oral cavity, and it gets activated into the stomach of pH 4 to 6. Do remember. Then we have gastric lipase. Gastric lipase, it is secreted from chief cells of the stomach. It is also activated in acidic pH, and pancreatic lipase, it is secreted from the exocrine portion. Now we have some of the characteristics which are common in lingual and gastric lipase. First, they both are activated in stomach. Do remember, the digestion begins in stomach, ends in small intestine. Both are activated in acidic pH. They are responsible for breakdown of medium or short chain fatty acids having length less than 12 carbon atoms. And then we have these enzymes. They are more functional in children, less functional in adults, and in patients suffering from cystic fibrosis. Got it?